Welcome back, guys. Last time we talked about mitosis and how it was we needed to replicate DNA to double that DNA so that we would have two identical cells at the end of mitosis that would be just like the original. And we talked a little bit about how that DNA could be separated, how DNA polymerase would be involved. And we really were just stuck with the genetic material, that DNA, and we didn't really talk about what was the purpose of that genetic material. We talked about how it had a basic definition of a blueprint for life, that it coded for something. Well, this is where we can kind of really dive into that in this particular clip, okay? So we have two basic terms. Genome, this would be all of the genes, all of the DNA sequences that we possess. Okay? Or you could talk about the genome of a soybean plant or the genome of E. coli. All of these different living things have a blueprint for life called the genome. Okay? Now, some are very complex. Some are super simple. Okay? We look at the HIV virus. It's not technically a living thing, a virus, but it has 9 genes. Very, very simple, right? We look at the human genome, and we believe, based on studies, that there's 20,000 or 20,000 plus genes within the human genome. So what is it all about? Well, it so happens that the genome determines the proteome. That's not a term you hear a lot of about in the news, right? You hear about genetics and genes and maybe even genome. So all got that first part to the word, but proteome would be all of the proteins that are associated with our living system. Okay? What we're going to find is that the genome is, to a great extent, determining that proteome. Now, we learned the functions of proteins, right? We learned that proteins were, in fact, playing many different functions. Some were enzymes, without which we wouldn't exist. Some are hemoglobin, without which we would not carry oxygen around the body very well. Some of which were structural proteins that gave a cell shape or connected to cells when it comes to desmosomes. Any number of things, right? Even pumps in the membrane or channels, that's the proteins are where it's at, okay? But here you see there's a connection between this blueprint for life and the proteins that we're producing. Now, here we have a very basic cell. We've got our nuclear envelope here, so this purple zone is the nucleus. Here we have the DNA, so we want to talk about the DNA and how it is we're going to use this DNA now, not to replicate it, but to produce something functional and something useful from that DNA. All right? Now, the way I like to think of this, and the analogy I like to use, we're going to use a couple different analogies, but let's take this analogy for example. We have a library on campus, or you might think of a library someplace else, but some of these books in the library are reference books. They are not to leave the library, right? You can rent it out for an hour. You can take notes from that book. You can maybe even take some images from that book, but you're basically taking notes, and you could go away later, leave the library, and utilize those notes in any way you see fit, okay? So this is a similar situation. Within the nucleus, there's a gene library. There's a whole set of sequences, set of genes. Some are very short, some are much longer, and these sequences of the DNA are genes. They're going to be coding for certain things. What we're going to find is that this DNA, the genes found within this genome, okay, will code for what we call mRNA. Now, they still end in NA, you notice, okay? So the DNA is your reference book. Can't leave the library with it. Can't leave the nucleus with this DNA, okay? The way this typically functions is we take notes from that DNA, and those notes are some kind of message that you've written down. mRNA, messenger RNA. RNA are those notes. Now they're going to be in a similar format. okay? And I like to think of it almost like you read this book in English and you took down notes in the same language, let's say, okay? Maybe it's just a shorthand version of what you read in that book. Still the English language, still nucleic acids. So we're sticking with the language of nucleotides. Basic ideas that nucleotides make up DNA and RNA. Now, this first step of the process we call transcription. When you transcribe, you're not changing the language. And in this particular step, we're using something very different than what we talked about with DNA replication. This is not DNA replication. We're producing a new molecule called RNA. So why not call it RNA polymerase? If I want to talk about, we'll put in a, we can talk about replicating the DNA. We can talk about DNA replication where this would involve DNA polymerase. But now when we talk about transcription and we're moving down to produce this mRNA molecule, well, you're not making a polymer that we call DNA, we're making a polymer we call RNA. Okay? Still nucleotides that make this up, but this is called transcription. So where does transcription occur? Transcription always will occur within the nucleus of the human cell. So we can take the notes there, and then we can leave the nucleus to perform the next step. Okay? Translation. When you think about the word translation, I hope you think, well, you might translate something from the English language to Spanish, right? Or backwards, the other way, right? Well, in this case, we have a language of nucleotides. That language, we call them RNA and DNA, okay? Now, this shorthand version of notes that we take from the DNA, this message, imagine leaving the library with some information you got from your reference book, okay? If you leave the library with those notes, you could go and use those notes to actually produce a product. Let's say you went to the library and you read a book on how to build an outhouse. I don't know why I thought of that, but it's what came in my head. So you read a book on how to build a house, right? And now we leave the library, and with those notes in hand, you can go and buy what you need from Lowe's. You can cut those 2x4s up and build yourself a house, right? As small as it may be, right, with an outhouse. Okay, so this is kind of what we're doing down here when we talk about translation. Okay, translation, we're going to change this message. So here's our mRNA. We're going to use that message, and from it, we're going to produce a polypeptide. Now, that's just a fancy word for a polymer of amino acids, okay? A peptide is a fancy word for protein. So, what are these little orange circles here? They are amino acids, right? These amino acids are going to be strung together in a certain order. We call that the primary structure of the protein, right? And then we know what that's going to do, right? We learned that proteins are going to function based on their shape, and this long chain of amino acids will curl up on itself and it'll make a very interesting shape that functions for us. This process of translation will occur outside the nucleus. The ribosome is going to play that role of a protein factory for us, reading the mRNA and producing a protein as a result. Okay? Now, in biology books, you're going to see the words, when they talk about this kind of stuff, they'll say central dogma. Big fancy, why do we have to make it sound so hard, right? But it is so central to our understanding of biology and how cells replicate that you're going to talk about transcription and translation all the time. I mean, I don't know how many biology classes I took, but, throughout graduate school, this came up all the time, right? It's just so important to our understanding of what's going on, okay? So let's stay with this idea. Let's look at what's going on then. As we go through transcription, what are we having to do, okay? So here you have an image, and this image represents at the top DNA. That makes sense, right? We've got 2 strands. Now this isn't all wound up like a spiral staircase. They've kind of straightened it out so we can see what we're dealing with. But you do have 2 strands. We got to imagine that there would be these hydrogen bonds existing between these different nucleotides. And this is going to be the DNA we start with. Okay, so here we have DNA, and let's just say that this is a particular sequence within the DNA that we're going to call a gene, right? Now, I don't want to replicate this DNA. I don't want to just pull this blue and gray strand apart and make double with a DNA polymerase. What I want to do is come in with RNA polymerase, go through what's called transcription to produce an mRNA. Okay, so let's think about what would happen then. If I want to produce mRNA, okay, and I want to copy these nucleotides at the top, and let's say that this blue part, this is the gene, okay? I want to just read that one side of this gene, okay, to make mRNA. All right, well, what I'd want to do is I'd want to consider, if this was all gone, right? Let's say we're imagining this is not even here. This is not the side that we're going to read as a gene. I want to read T, A, C. I want to make a copy of this, but I want to take shorthand, right? We're making mRNA instead of DNA, okay? So for every T, I would want an A. That's the same rule we saw with the DNA, right? Adenine is always going to bind to these thymines, okay? And adenine's still going to be the one that we add to a thymine when we make RNA. Let's look at this one over here with the C and the G. Okay, for this cytosine, okay, well, we always put guanines on those. So when I make the message here, I'm going to use guanine, right? So there's a G there. But what about this one I just skipped, right? A's bind to T's. We learned that. A's bind to T's. That is a DNA thing where those are always bound. But there is a slight difference when it comes to this mRNA. Remember, we're going to use shorthand, and part of our mRNA shorthand that makes the nucleotides different is that uracil is going to be used in place of thymines. Okay? So in my production of these notes, this mRNA transcript, we call it, because it's transcription that it's going through, I'm going to put A's where there's T's. I'm going to put U's where there's A's, G's, C's, A's where T's, right? So basically anywhere I see an A, there's just a little bit of a twist on it, right, when I make an RNA molecule. I mean, anytime I see an A, I'm going to attach a U to those. And that's why you see this difference down here. Think of it like shorthand. It's just a little different. We're taking English language in our textbook, and we're still going to have English language, but it's kind of a different kind of notes version. All right, so basically what we're doing is we're taking these down one at a time to produce all of these nucleotides in our transcript, in our mRNA. Now this message is going to leave the nucleus, and then the ribosome is going to determine which amino acids will correspond, which of the 20 amino acids correspond with this code. Okay, so really in the end, the reason for this code is not so we can copy it. That's just a waste of energy, right? If I had a blueprint and I just had a blueprint there I constantly copied and never used to make anything, it's kind of a useless blueprint. So here we see where this actually comes into play. The code of the DNA determines the code of the RNA, and the code of the RNA determines the amino acids. How it determines that code is a little bit more complicated, though. It so happens that every 3 nucleotides resulting in 3 nucleotides of RNA determine 1 amino acid. Okay, so we talk about triplets of DNA. TAC is a triplet. The next triplet, TTC. This is our gene, okay, and it comes in 3s. Next triplet, AAA. What are we going to get as notes, as a message from this blueprint? Well, each triplet codes for what we call codons, right? So triplet number 1, TAC. From that, I got an AUG. That's our first codon. So happens, this codon results in a very specific amino acid. Now, I'm not going to ask you to know all of these names of the amino acids. It's good enough that you know there's 20 of them and that we can string these together in many different orders, right? Our primary structure. I just want you to realize that triplets code for codons, and then the codon in the mRNA result in very specific amino acids. And this is why the DNA is so important. Okay? This is why the code, the specific code of that DNA is important. And that changing that code would result in a change of the code down here where we see our codons, and may very well result in a different amino acid. Big whoop, right? So we have different amino acids. What do we care, right? But remember, these amino acids have different charges, and they're going to wind up being quite possibly different shapes that could change function, right? So this is very key and very important to understand that DNA codes for RNA, transcription, RNA codes for protein, translation, and the code is very important to the resulting product and the function of that product, right? All right, so here we have kind of just some images depicting the transcription process, all right? So here we have the transcription going on up here in the top. We have an mRNA. I'm going to add the M here. And what's happened here is they pulled the 2 sides of the DNA apart. And in this case, we didn't pull it apart to make new DNA. We had to read this side of the DNA. It looks to me like it got started way over here somewhere. This was the beginning of the gene, and probably this was open up way over here. And then this RNA polymerase, right? We're not making a string here of the DNA nucleotides. We're putting a string of RNA with these U's, right? So here's our string, okay? And then we remove those from the nucleus, go out into the cytoplasm where we see our ribosomes. Now some of these ribosomes are stuck to some membranes out there, right? The rough ER, and some are not. But what happens is this ribosome will just march along. It's going to read the first set. Okay, here we have AUG. Well, there so happen to be other molecules, like this one here, called tRNA molecules. And out in the cytoplasm, these are little carriers. They're going to carry amino acids around for us. So if AUG codes for methionine, well, guess what? That's what this particular tRNA's job is. It carried in methionine. Okay? Okay, we're making a string of pearls here, right? We're just kind of attaching them together. We're making this long chain of amino acids. Okay, and then this ribosome marched over to the next set. Here we have our next set. And this tRNA's job was to bring in this thing called phenylalanine. Again, don't worry about the full name. This was an amino acid, came in first. This was the amino acid that came in next. And a covalent bond, a peptide bond, formed between them. What's going to happen next? Well, it's going to march to the next one. Okay, this ribosome is just kind of ratcheting 3 at a time down this long code. Okay, and it's going to have the next tRNA that's going to bring in arginine, just another amino acid. So guess what? The next one that's going to be tacked onto this chain will be arginine. It's going to just keep on building this string of pearls. Okay. Before you know it, you're going to have a long chain of amino acids, and you're going to have a functional protein. And so this image kind of shows you this kind of idea, right? Here we have a ribosome. That's a protein factory. And it's going 3 at a time. Go down 3, read the next amino acid. Go down 3, read and bring in the next amino acid. Down 3, and these tRNAs are bringing each of the amino acids in as we go. Now, you could have one message out there in the cytoplasm and lots of ribosomes reading that same message. So it's not like the protein you end up with, they say polypeptide, but let's just call it a protein, okay? You can make not just one protein from this message, this mRNA, you can make several hundred, right? Or thousands of these if they're needed in that abundance. Now, what's the big deal about the code? Let me just really hammer home. What if the DNA code changes? Well, it's going to change the RNA code, right? And then that could change the amino acid that's brought in to build our protein. So let's look at this as an example. Okay, sickle cell. Sickle cell is not a good thing, right? It's not a good thing to have. It's a disorder. And it's a genetic mutation is what it is. A mutation would be any change to the genetic code. Now, some mutations can occur, and they may have very little effect on the biological system. So I think it's a misunderstanding that people sometimes come in with, thinking that mutations are all bad. A lot of the mutations that get our attention are absolutely bad. And that's why there's a misconception there. Mutations occur, sometimes we never know it. But this particular mutation, and here they have an example, okay? So this is normal. On the left side, we have wild type. You might as well put normal in there, okay? The normal functional gene that produces hemoglobin has a certain code when you look at the DNA, the gene, okay? And in part of that code, it says CTT. That's the order. And the CTT codes for what? Well, C's have G's, T's have A's, T's have A's. So our mRNA is right down here. This is our transcript, right? This is transcription going from the normal DNA to this normal code mRNA resulting from it. And we get a certain amino acid as a result of this set of notes. The GAA codes for something called glutamine. Okay? And we get a nice protein out of this, a nice hemoglobin subunit that will allow us to function and to have normal red blood cells with the ability to carry the oxygen around our body. Okay, now let's go back and consider the mutation that is the sickle cell mutation. In the case of the gene for a mutant hemoglobin DNA gene, we don't have CTT, we have CAT. All right, well, what's the code we're going to get from that? Our transcript, when we go through transcription in the nucleus, is going to result in a G for a C, a U for an A for a T. Now, this results, it just so happens, when we have translation go on down here at the bottom, at the ribosome, we're going to end up with something that we refer to as a sickle cell hemoglobin protein. A very different amino acids brought in, and it's so different that it results in a different shape to hemoglobin. A shape that causes some major pain if you have sickle cell and can be life-threatening. All right, so this is why we worry about mutations, and these mutations occur for any number of reasons. Why do they occur? They occur when there are mistakes in replicating the DNA. That happens. They occur in the presence of mutagens. Some of them may be environmental. Some of them may be the incidence of the sun, right? So UV rays from the sun can cause mutation. X-rays can cause mutation. So when you get an X-ray of your leg, right, if you're a younger person, they'll cover up your private area, right? Because in that region, the last thing you want to have is mutation that would result in possibly mutation in sperm or egg that you would produce. So here's why it's so important that we understand mutations, understand the importance of the code that ultimately results in a protein. Okay? All right, we're going to leave it at that here, guys, and then we'll talk about a little more about genes, about heredity, and how it is that we can carry genes without actually showing them on our outside. Okay? I'll see you guys next time.